Okay, well, hello everybody. This is the final uh, presentations for the activity metabolomics and mass spectrometry short course that's turned into somewhat of a short course workshop and lecture series. So you can choose whatever one you like. This one is going to be a little bit more of lectures or presentations on activity metabolomics. And we're going to try to do something today that we've been told by our IT department it's not possible. So, and that'll be towards the end of the last presentation. So I think we can make it work, but we'll find out. We'll find out very shortly. So actually, the Scripps Research Institute has been around now for 100 years, and it was formerly known as Scripps Metabolic Clinic, and it gradually evolved into what it is right now, which is doing many different types of research, chemical biology, chemistry, pure chemistry, molecular biology, immunology, and a host of other different types of fields that Scripps is actively pursuing. And I've been at Scripps now I'm in my 35th year. So I've been here for roughly a third of Scripps's lifetime. And I have to say, it's actually been very enjoyable from many different perspectives, extremely stimulating, very open to doing different types of work. And it's just been a great place to have spent the last 34 years. Here's a picture of a part of the Institute, a dedication memorial to Bernie Galula, who was the first dean of the graduate school at Scripps. And for those of you who attended the previous meetings, this is where we're hosting the meeting today at the faculty club here at Scripps Research. Now, throughout the past 4 weeks and the previous 3 weeks, we've had a lot of different speakers from within my lab, including Corey Wang, Elizabeth Billings, Myrna Tobia, Phil Webb, Winnie, and Lynn. This week, I'm just going to be giving a short presentation on some of the original work that we did to get us started in metabolomics. And I'm going to fast forward to last Friday to describe something that just came out, which is kind of a culmination of a 2-decade effort. So you've heard us discuss a wide variety of topics, including in-source fragmentation, phantom metabolites. Actually, that was a topic that came up with respect to Martin Guerra, who was a colleague of mine who's going to be talking today, who was also the primary author on that paper. We've talked about mechanisms of diseases with John Yates, population scale metabolomics with Juliana Ivanovic, and digital biology, and a variety of things that is playing a role in that with respect to metabolomics, including similarity searching, in silico identification, in-source fragmentation, and digital biology. Today, we're really focusing in on the application side of the technology. And given that, I'm not going to be talking about this at all today, but I just wanted to show a couple of examples of activity metabolomics being applied and pulling up molecules that has turned out to have some really interesting biological relevance. One of my favorites, I'm going to show 2 favorites right now. One is neuroprotectin D1. This was discovered by Oscar Llanes, and it was a paper we published in Chemical Biology, Nature Chemical Biology, over a decade ago. And it turns out this particular molecule, what Oscar found was it had the ability to initiate the differentiation of stem cells. And that was just a small part of a wide variety of discoveries that Oscar made in that particular paper. It was really just a really incredibly interesting, at least to me, interesting paper. It basically described how the plasticity of the stem cells could potentially be at least partially explained by them being rich in highly unsaturated lipids. And through enzymatic reactions, and the stimulation of those stem cells through the differentiation process, those highly unsaturated molecules become the scaffold for a vast variety of other metabolites that make up the more mature cells that the stem cells differentiate into. And perhaps my all-time favorite, and I mentioned this last week, was 3-indole propionic acid. 3-indole propionic acid was a project that was initiated by Pete Schultz, who's now the president of the Institute. This was back in 2009. And Pete was looking at mice with and without gut microbiota. And one of the more interesting discoveries we made in that particular paper was that 3-indole propionic acid did not exist in animals that didn't have a gut microbiota. And in fact, it was further determined that the specific bacteria that was producing it, and so we all have, or the vast majority of us have this particular molecule, and it's this 3-indole propionic acid, or 3-IPA, and it does come from this particular bacteria, Clostridium sporangiis. This was a discovery that was made. Bill Wyckoff, who was a postdoc in my lab. Oscar is now a professor in Spain. So with that, today we're going to have 3 separate speakers, including Martin Guerra from Leiden University Medical Center, and he's going to talk about going from endogenous to the clinic. And Martin is very active in this area of the discovery process of endogenous molecules being biologically active. And he's actually going to have one example where he's taking a particular molecule all the way to the clinic. Marcus Rinchin is he's actually one of the few MDs that I've had in my lab, and he's since gone on to Aarhus University in Denmark, and he's been, as you can imagine, as an MD, he's been very much involved in the medical side of using both metabolomics and proteomics, not only to understand disease, but also to also modulate the phenotypes with metabolites. And the last speaker is Shua Han from Duke University, and she is going to talk about gut microbiome bioactive metabolites. And she previously worked with Justin Sonnenberg at Stanford University. And I showed this in my first lecture. This is basically the whole premise behind activity metabolomics, where it's nothing new. There's nothing new about this. I mean, we know metabolites act on the DNA, they act on the RNA, they act on proteins in a wide variety of ways. The only thing I find interesting about this is the technology that is now driving the discovery process, and that in activity metabolomics, we're largely, to a large extent, using mass spectrometry to identify molecules that are significantly dysregulated, and then identify what those molecules are and look for their specific biological activity with respect to a specific problem. Like in the case of Oscar, that was stem cell differentiation. So I'm just going to spend a few minutes discussing a couple of different things that I haven't discussed before, and also a few things intermingled in there that I have discussed before. I mean, our lab is largely driven by technology, and in collaboration with biologists here at Scripps, we will tackle biological type problems with the hope of not only solving those biological problems, but also helping us further develop the technology. And over the years, we've had a wide variety of different types of papers. These are probably some of the key papers in those efforts. And largely, the primary goal in all of this work was to identify biologically active metabolites. And then, so the first study we actually were involved in was a sleep study. This was back in 1994. it was initiated by the former president who recently passed, Richard Lerner. Richard was not a metabolism person at all, so I think it really speaks to his incredible diversity in terms of the types of science that he got involved with. He was just a really brilliant scientist, and I don't use that word very often, but he truly was a brilliant scientist that we were lucky to have at the helm of Scripps for a couple of decades. Also part of the sleep study was Ben Cravatt, who's now a professor here at Scripps, and he's gone on to develop a field called activity proteomics, or proteomic activity profiling. So in this sleep study, we were looking at cerebrospinal fluid, or molecules that were dysregulated in animals that were sleep deprived. And we did see a whole family of molecules, particularly a set of lipids that were actually significantly dysregulated. We used a wide variety of analytical techniques that were available to us at that time in 1994. Tandem MS, not at high accuracy tandem MS, but just this was on a triple quad instrument. Accurate MS with M over Z was determined from a magnetic sector instrument. And then we did a wide variety of HD exchange experiments. We examined the isotope patterns. We did preparative LC for collection of the metabolite for NMR analysis. And ultimately, Den, who was really the head leader in this particular study, Ben Cravatt, he synthesized an authentic standard and then compared it to the molecule that we thought it was. And ultimately, we did identify it as oleamide, relatively simple fatty acid amide. On the positive side, the oleamide did induce a sleep-like state in the animals after 4 minutes, so it did have the biological properties that we were interested in mimicking. The downside was that it took 8 months for us to actually identify this. So that was really a major limiting factor, at least for me, in terms of how much effort I could put into this. And it actually incubated in my mind for quite a few years as I tried to come up with ideas on how to potentially solve the problem, which was a need for more effective means of statistically assessing these molecules and identifying what they were. One of the first solutions that actually came in the form of XCMS. XCMS was developed by Colin Smith, he just came out of as an undergraduate at that time. He came into the lab and I asked him to help us solve this particular issue of doing statistical assessment of the LCMS data. And on a Saturday night, I got an email at around midnight. And he said he thought he'd solved the issues that we were having with respect to doing accurate comparative analysis. And Collins, as a kind of a tribute to his solution to this, this has now become a standard approach that is used in pretty much all the instruments that all the software that's developed for LCMS comparative analyses, especially metabolomic experiments. And his approach was basically, and he's now a professor at Wesleyan University, and his approach was a nonlinear alignment of the data based on the metabolites that were inside of the samples, and he consistently across the different samples, and he used those molecules to align the data with not internal standards, but the molecules that were inherently in each of the samples. And that nonlinear alignment ultimately allowed us to do a much better statistical assessment. And it's why this sort of approach in various forms is still used today. That allowed for a statistical assessment of dysregulated peaks, pulling out the molecules of interest. And the next task was to identify what those metabolites were. I should mention that Ralph Tottenheim in 2012 created an online version of XCMS, which has become very widely popular. And now it hosts, in addition to Metlin, over 55,000 registered users. I think, obviously, the population of metabolomics people is this seems excessively large. So I suspect a lot of people outside of the field are connecting onto this as well, because we do offer a data sharing aspect to this. So these are not just metabolomics people per se. From the identification perspective, actually, Winnie went into this in a great deal of detail, so I'm not going to talk about this very much. It was built in the early 2000s to facilitate molecular identification. It has the M/Z data that we're interested in just as an initial filter, and it also has the MS2 data. And here's the MS-NS data from the oleic molecule that I mentioned earlier in the sleep study. So now we're at around 936, close to 936,000 molecules. And I'm just going to reiterate what Winnie pointed out last week, which was there's been a lot of individuals who've been contributing to this. And I really like to point out that Winnie and Aries Asporna are the primary contributors to this now, because they've now added over 98% of the molecules, molecular standards with MS-NS data in Metlin are due to both Winnie and Aries. And we have neutral loss data in there now, and we also have ion mobility data in there right now as well. So it's growing in many different ways. Okay, so this is essentially all I wanted to talk about, except one last thing. So we've been working on this, obviously, for many years, since the early 2000s, trying to come up with solutions to the identification process. And finally, after, and I have to say, this is not in its final form. This is where we're still working on improving it. And what I'm going to present to you right now, very briefly, is a fairly truncated version of what we ultimately want to put out. And it did come out. I saw it for the first time last Friday. It says that it came out on Thursday, so I may be off by a day. So, but it did just come up. And this was a combination of XCNS Metlin, allowing us to do both the statistical assessment and the identifications all within one package. And the current demo version, which does not have all the features that we want to have in it at the moment, is publicly available. Actually, we've had actually a great deal of people come in and demo it, and actually I got some feedback from a former postdoc yesterday, Carlos Gieres, sent me some of the ideas he's been making using the demo version. And interestingly enough, it's not even from a QTOF instrument. It's coming from an LCQ type of instrument. So it's not even an ideal type system, but he's getting really good, solid ideas from it. So obviously, this is pretty exciting for us in terms of finally combining something that will help facilitate the identification process using really relying solely on molecular standards. And I think the last point I want to make is in this particular figure that's in the paper. And I mentioned this last week, but I think it's worth reiterating. Obviously, we're starting off with a huge number of molecules in the database, and that number of molecular standards with the MS-NS data on all of them, you could think as being a fairly daunting process to get to the point where you start with that, you give it some data, and get down to the actual identification. It turns out that it's not that daunting at all. It's actually extremely straightforward. Basically, one of the big filters that helps narrow down these molecules very significantly is the precursor ion filter. Basically, the precursor ion filter typically gets us well below 100 molecules. And then when we add the MS-NS data for comparative analysis, we're typically at roughly 2 molecules. So I guess the bottom line for me in terms of the development of XCNS Metlin is that what we're trying to do is use a very experimentally based approach, or standards-based approach to making these identifications and not rely on speculation in terms of what these molecules might be based on similarities, which we know has issues. So with that, I'm going to start. I've already introduced Martin, give you a pretty good sense of how much of an effort we've had with him over the years. And just this past year, in source fragmentation paper we just had in Nature Metabolism, the recent paper that we had on tandem mass spectrometry across platforms, and a whole host of other papers that I've had with Martin over the course of the past couple of years. It's really been a pleasure working with him. But of course, he has a huge effort going on at Leiden University Medical Center, and his efforts with me are relatively small in comparison. So with that, I'm really happy to introduce Martin to talk about endogenous to the clinic. And Martin, hopefully you finished dinner and you're ready to present.
Thanks, Gary. Thanks for the invite. It's my honor to be invited by one of the godfathers of metabolism. So,
hold on, Martin.
Should I pull up my presentation?
Yeah, sure.
All righty. Can you see it? Because I cannot see you guys anymore.
Yes, we can see the presentation.
All righty, perfect. So yeah, thanks for the kind introduction, Gary. And I think, yeah, it's a perfect introduction to my work. I'm a pharmacist by training, and as you see here, I tend to bike when I'm not in the lab, and I also bike a lot with Gary. And I promise I'll tell an anecdote. We'll get to this in a second. But so the question for me personally, evolving and developing myself in the field of metabolomics and also lipidomics has always been, so what do we make from it? Where do we use it for? And I was involved in a lot of analytical developments throughout my career. But at one point you start to ask yourself, so we developed all this great technology like Metlin, like AMMS Metlin, like many other things. What are we going to use this for? How are we going to apply this? How are we going to leverage this? And I think this is at the core of my career, I would say, at the core of my work. And I crammed 15 years of my personal work into 20 minutes of presentation, so bear with me. It's all about lipids. I'll introduce that briefly to you. And the attempt of moving things forward from technological preclinical development into the clinics and to really make use of it for the patient directly in the end. Gary has shown this already, so maybe the anecdote where activity metabolomics starts is in Gary's former car, which was called Osito, and we just came back from a bike ride to Palomar Mountain, and I think then the idea was
Martin,
born. Yes.
Sorry, we cannot see the slides.
Oh, thanks, Marcus. Then I stop sharing, and I'll do that again.
But we could see the slides.
They just weren't moving.
I don't think you had it on slide preview, or you had it turned it on.
Can you see the slides now?
Yeah. Yes. Yeah.
But they're not
There you
moving.
go. Yeah, just click on the
Marcus,
next slide mode. Okay.
we're at activity metabolomics.
No,
Rich
it's
net-
on the final slide.
You haven't started your slide presentation.
I did, but then I have to share the full screen, then it usually works. I do it like this, and now I go here, and now it should work. Yes?
No, we just see you.
Working now? No?
No. Before we could see your screen.
Yeah. Share. Okay. But now it should work.
Just
Click on the
There you go.
There you go.
There you go. Yeah.
Okay, there we go.
Yeah, finally.
Yes.
It's great.
So let me get back to where activity metabolomics started. In my opinion, it started in Gary's car coming from a bike ride to Palomar Mountain. And I think we started to discuss, yeah, so what is needed in metabolomics? Where is the field going? Where would we like to have it moving? And I think we discussed for several hours. And the final essence was really that, we started to think about the concept of how do we identify bioactive metabolites? How do we tear them apart from, let's say, the metabolic noise, if you want to say so, and how do we make use of them in the end? And you have seen this develop and this idea develop in many fields. Also thinking about microbial metabolomics, the interaction between the microbiome and the human physiology. Now, Marcus picked up on this when he was a postdoc in Gary's lab, and together we wrote this wonderful paper, which I like to use as my starting slide, just to reiterate where we're coming from and how data in a cell, if you want to say so, information in a cell is translated, transformed, and also to make the point that if you would have drawn this slide 20, 30 years ago, it would have stopped at the protein level. And nowadays, metabolomics and lipids, metabolites, have become an integral part of the central dogma of biology. And this is also an important realization that has been taking place in the last 20 to 30 years. Now, the other important point, and Gary, Oscar, and myself, we reviewed that for cell metabolism a while ago. The other important realization is that this increasing biochemical knowledge has been going hand in hand with technological developments. So you cannot tear this apart. And a fun fact here, maybe the cradle of metabolomics is actually in the city of Leiden, where a chemist called Boerhaave isolated and identified and characterized the first metabolite ever, urea, from horse urine. So he basically took 100 liters of horse urine, boiled it until it became some sort of cream, and then he thought it would be a good idea to store it in his cellar for a year. And surprise, surprise, very large crystals formed, and that was actually urea. So in the city where my lab is located, it all kind of started some 300 years ago. Now, having said this, I want to switch gear a little bit into the realm of lipids. So lipids, well, obviously lipids are also metabolites. Their definition is still a little bit vague. Even the UPAC doesn't really know what to make from it. And even the UPAC calls it themselves a vaguely defined term for metabolites soluble in organic solvents. So water insoluble metabolites, if you want to say so. Now, this slide is just to show you that if you look into a cell and you look into the different compartments of a cell, you will find that all compartments of a cellular system show very specific lipid compositions. So, for example, mitochondria have cardiolipins as specific lipids. The endoplasmic reticulum is the hub for sterol biosynthesis. Lysosomes have very odd kind of lipids called BMP lipids. Plasma membrane is mainly made up by phospholipids, and all have very significant structural differences and a very specific lipid package per organelle. Now, this realization has also led to the realization, aligning with what I said earlier, that metabolites, lipids have become an integral part of biology's central dogma, and as such, they can shape biological phenotypes. They can even shape cell fate and cell fate decisions. So, depending on your lipid composition, you might go into fate A or fate B. To underline this a little bit for you and give you maybe a little less abstract example, I picked one of our recent publications where we investigated lipidome and the relation of the lipidome to the biological phenotype in T cells. If you roughly divide T cells, you can have pro-inflammatory T cells like conventional T cells, cytotoxic T cells, T helper cells, helping to kill intruders, and you have regulatory T cells, anti-inflammatory T cells, suppressing immune response. So the first question we ask, so do these cells now really come up with a different lipidome? And yes, indeed, it's vastly different. And it's shown up here in the PCA plot. So we did several lipidomics analysis, monitoring several thousands of lipids actually. And you see that even in the unstimulated states already, the triangles and the circles, conventional regulatory T cells, are vastly different. That is just shown here again in the volcano plots. And when you stimulate them, they also show a different modulation depending on the T cell type. So again, there is a very clear-cut link between this lipid composition, the molecular composition of these cells, and their biological phenotypes. If you then looked a little bit deeper into this, you could mill out some details as shown here. We don't have to go into the details, but regulatory T cells, so the anti-inflammatory T cells, they showed a much higher unsaturation level of these lipids, so many more double bonds in these lipids, which aligns very well with other fields in immunology. Just think about docosanoids, anti-inflammatory mediators, specialized pro-resolving mediators, eicosanoids, and so on. So anti-inflammatory signaling lipids come from unsaturated fatty acids, and we saw many more of these in regulatory T cells. The next question we ask is, so is this potentially differentially fed? So what is fluxing into this composition? Where are these lipids coming from? How are they made? Are they made differently, or are they taken up? So we investigated by 13C isotope tracing. And again, to just cut a long story short, otherwise I need 20 minutes only for this story. The essence of what we found is that conventional T cells, they really like to eat glucose, and they use it to produce triacylglycerols and short saturated chains. Surprisingly, regulatory T cells, that has not been shown by us, but by others, they really love to eat lactate. So the ugly duckling of metabolism, where we always thought that's kind of a waste product, is really turning into a fuel. And then I think Josh Rabinowitz wrote a very interesting review about this, that this is also conceptual change just taking place. So Tregs, they really love to eat lactate, and they use it to make very specific lipids, helping them to fulfill their functions. Now, but taking this a step further, when we think about the T cells, then you can already think, yeah, so if I have a tumor microenvironment, I have a tumor, which usually has a Warburg effect, right? Producing a lot of lactate. Regulatory anti-inflammatory T cells can live in this surrounding. They even love to eat lactate. Not a good thing, right? Because you don't want to have the Tregs proliferate, but in tumor microenvironment, you want to have the pro-inflammatory T cells to proliferate. So this already shows you also a link into the bench to bedside story. So how can we further leverage this? And it's just a couple of examples where lipid biology targeting drugs are being leveraged. And I will dig into one of our fields of research where we target the enzyme dehydrocholesterol reductase 24. Now that actually starts with my PhD in 2005. And when I started my PhD, my supervisor said, this is cholesterol biosynthesis. I want you to make an assay so that we can study drugs and their selectivity and IC50, blocking all the different enzymes within this pathway. Then I didn't see him for half a year or a year. but that doesn't matter. I made the essay, and I think we nicely published it, and I will just quickly explain to you how we did that. So the thing with cholesterol biosynthesis, it all takes place in the endoplasmic reticulum. So it's very hard to isolate these enzymes because they're membrane bound. So what I came up with was a whole cell assay. We then incubate with test substances, and basically, to cut a long story short, we did the GCMS analysis, and by spectral matching, we then identified the accumulating sterols, and deducing from these patterns we get, we could identify one or several target enzymes within the pathway, and then making use here already of carbon labeling, we could construct IC50s by defining percent inhibition. Now, we then applied this, and I synthesized a couple of inhibitors myself, and we closed a lot of gaps where there were basically no inhibitors or no selective inhibitors for the different enzymes in the cascade available. We made the first selective inhibitors for latosterol oxidase, delta 8-7 sterol isomerase, oxidosqualene cyclase, 70-hydrocholesterol reductase. Now, 15 years ago, I have to tell you, there was not a lot of interest in these compounds, and it was quite hard for us to publish a lot of this stuff. Luckily, this changed, and just this year, there was a back-to-back publication of 2 papers in Nature showing that actually one of these intermediates of cholesterol biosynthesis is a regulator of ferroptosis. Now, I'm not going into the details. Look it up, 70-hydrocholesterol. Very interesting story where Nat Porter spent a lot of time on. Is surprisingly the fastest oxidized lipid in nature. And as such, it controls ferroptosis. Nevertheless, just to show you, there has been, also here, there has been a change of mind or a realization that cholesterol biosynthesis and lipids in the precursors, it's not just about making cholesterol. There is much more biology going on in the back that we didn't think of. But well, also maybe it's not that surprising. Why would you have such a long biosynthetic pathway if the intermediates basically are useless? Nature is usually quite efficient. And so, but the one paper or the one inhibitor class that we moved forward, they were aimed at blocking the HCl24, the hydrocholesterol reductase 24. Now, first we didn't really know what to make from it, but then came this paper actually from one of Gary's colleagues at UCSD, Chris Glass. And Chris, he investigated foaming macrophages. Foaming macrophages play an important role in cardiovascular disease. So everyone thought, these pro-inflammatory cells, they gulp up the lipids and they become super inflammatory. Now, surprisingly, Chris found the exact opposite. And what he found is that when these macrophages accumulate these lipids, they block cholesterol biosynthesis at a very specific spot, namely at the HCl24. Then they accumulate the precursor of cholesterol. Be aware, please, the devil in the detail, one single double bond difference here in the side chain. This accumulates, and this now exerts anti-inflammatory actions. So they are intrinsically anti-inflammatory, these foam cells, rather than pro-inflammatory. Now, what this desmosterol does, what the pharma industry tried to accomplish for 20 years, is to selectively activate the liver X receptors. There's no doubt about that this could be a cutting-edge therapeutic avenue, but no one has succeeded in tearing apart activating SREBPs from LXRs. And SREBPs boost triglycerides, so hypertriglyceridemia, and that's a side effect that killed most of the drug development projects. Now, having seen this, we thought, well, we already made inhibitors for these enzymes. They're very selective. Let's take them out of the drawer again, optimize them, and move them forward with basically this concept. We block the HCl24, we accumulate an endogenous bioactive metabolite named desmosterol, which is a selective inhibitor, activator of LXR, exerting all the positive effects while even blocking the SREVPs. Now, first paper came out in PNAS 2019, where we did the fundamental work in a peritonitis model, actually showing that this holds true in vivo when we use our published inhibitor SH42. We can block the enzyme, we boost the levels, and this funnels into an accelerated inflammation resolution by a change in lipid composition and a change in cellular phenotypes. Now, then we ask ourselves, yes, so what are potential indications where we can use this? And yeah, MASH, nowadays MASH, formerly known as NASH, was kind of a no-brainer because we had anti-inflammatory effects. That's one hallmark of NASH. And we had the lipid-lowering effect by blocking cholesterol biosynthesis and activating LXR. That's another hallmark of NASH. Now, basically, we moved into this area, and this is just a progression of NASH shown here. So it's non-alcoholic steatohepatitis. So these people are not heavy alcoholics, but still they develop, likely by overeating, by a bad diet, too much liver fat. This causes ultimately liver inflammation and processes into cirrhosis and cancer. Please be aware, NASH will be the predominant cause of liver transplantation in the Netherlands by 2026. There is no treatment. The only treatment is liver transplantation. It is estimated to have several 100 million of patients in 2030. This is a huge thing. So then we thought, okay, we use the mouse model, right? We started in a prophylactic setting. My apologies for the busy slide. If you want to have the real details, it's published in EMBO Molecular Medicine. And indeed, you know, we could show also we have target engagement. We can boost these levels of desmosterol in vivo. And to cut a long story short, we saw all the positive effects we had hoped for. You see the histology here with the oil red O staining, for example, and microlipid droplets here in the H&E also. So you have much cleaner livers. We block the accumulation of this liver lipid droplet accumulation. We have a reduced steatosis score, lower oil red O positive area. So overall, super positive effects, anti-inflammatory, and we are pumping out the lipids of the liver. Then, of course, well, here also you see this. You see this immune phenotype in more detail. We reduce these crown-like structures, which are one important marker of NASH. We saw more Kupffer cells, but intriguingly, these Kupffer cells were less activated. So we had more Kupffer cells, but these Kupffer cells had a different phenotype caused by this endogenous lipid, being in a much more resting state. Now, then, well, the referees ask us, okay, so you guys say this is LXR driven, show it. Do everything again in a liver and in LXRKO. It's only a 12-week mouse study, but no, 20 weeks, it's only a 20 weeks mouse study, but yes, we had to do everything again with an LXRKO, and just to cut a long story short, in the LXRKO, all effects are gone. Then we moved into a therapeutic setting. So we said, okay, if you already have a fatty liver, can that still be beneficial for you? So we put the mice 10 weeks on high-fat diet. They develop enough of these, a fatty liver, and then we started to treat. And we kept on feeding them on the high-fat diet. And here again, I mean, we could not reduce the steatosis, which had already developed during the first 10 weeks. But we again had a very clear-cut anti-inflammatory effect, as shown here, for example, also in AST and ALT levels. So very important markers for systemic liver inflammation. And again, crown-like structures heavily reduced, as well as F480 positive areas. So we could also translate this finding into a therapeutic setting. We're now working on leveraging desmosterol as a potential biomarker in patients with NASH. One of my PhDs is working on this. The final point I want to make here, I think our brain, and that's also this thing with NSAIDs and cyclooxygenase inhibition, we always draw correlations. So you see desmosterol potentially going up in a NASH patient, and then you immediately think, oh, it has to be something bad. Be aware and always potentially remember yourself, maybe it's a counter-regulatory mechanism and just your body or the system reacts to it, trying to compensate for something bad. So going up something with disease severity does not always mean it's also bad or it comes from a disease severity, but it could also mean it's a counter-regulatory mechanism that you potentially can leverage, as I have hopefully shown here. This just summarizes the whole thing. That's the molecule we use to block DHCR24. We increase desmosterol, we activate LXR, and we, in vivo, in a therapeutic setting, in a humanized mouse model of MASH, we changed cellular phenotypes and we caused anti-inflammatory and anti-steatotic effects. Now, the final example, we're working on more indications, but that's work in progress. I can't show you that data yet, but Grace has shown this in foaming macrophages, but we were wondering, or well, not wondering, but we kind of stumbled into this more or less, realizing that this might be a much broader physiological mechanism than we would have believed. So Laila and Dan from the Dutch Cancer Institute, they came to me with beautiful data on a novel lipid-laden macrophage phenotype they found in the tumor microenvironment of glioblastoma. And they had a lot of lipid signatures. So they came to me and said, well, can you help us make a sense of this and also do some lipidomics on it so we get a better understanding? And when I saw their data, I said, well, I think I know how that works because we're actually working on exactly the same thing. So the paper has just been published, I think 3-4 weeks ago in Cell, if you're interested, but it's the exact same mechanism taking place here in the tumor microenvironment in glioblastomas. So these macrophages come in, they gulp up the debris, they accumulate a lot of lipids. Again, they switch off the DHCR24, they increase desmosterol, they activate the liver X receptor, and they become actually anti-inflammatory macrophages. Now, the bad thing is that they kind of recycle these lipids for the tumor. By upregulating ABCA1 and ABG1, they pump out these recycled lipids, and the tumor gulps it up and uses them for proliferation. So basically what we stumbled across here is a sort of a vicious circle driven by a lipid phenotype. This is just some accompanying data showing you that indeed we can measure this on the transcriptome as well as on the metabolite level. I think my time is up. I'm a little bit over. My excuses for some hiccups in the beginning with pulling up my slides. I hope I could convince you guys that understanding lipid composition, lipid biology might be key to develop future therapeutic strategies and to tweak biological phenotypes. I think Gary has been reiterating this quite a lot, so I quickly go over this. Be aware of in-source fragmentation and have a look at AMMS Metlin. I think Gary will be very happy if you get some feedback from users out there. And with this, I thank all my collaborators and my group and you guys for your attention. Thanks.
Thank you, Mark. Yeah, that really convincing talk about you looking at the lipid dysregulation. I really like your final statement about the lipids. If they're upregulated, it doesn't necessarily mean they're bad. They're just trying to compensate for something that's going on in as you described with the NASH. So we're in the for time purposes, we're going to jump right to Marcus. So please unshare and thank you again. That was excellent. And Marcus, so are we all set with Marcus?
Yes. Let me just
So
turn on the
please share.
camera.
Marcus, I've known for over a decade now, and he came to my lab as a proteomic specialist. He's also an MD, and he was very interested in kidney disease. Since then, he's really expanded his efforts quite dramatically. Not only has he got involved in metabolomics and proteomics, but he's gone into he's in addition to kidney disease, he's looking at cross organ talk and disease modulation. And thanks again, Marcus, for agreeing to this presentation. I'm really looking forward to hearing what you have to say.
Hi, everybody. Thank you so much for the invitation. I hope you can see my screen now.
Yes,
Is
we
that
can.
the case? Awesome. Great. So yeah, I talk a little bit about our applications of activity metabolomics to maybe more complex medical settings. I hope it works. Yes, because I would start with this, that most of us and most of humans in Western civilizations at least have problems with a really sedentary lifestyle, right? So we have issues with, unhealthy diets and, together with genetic factors, this leads to really an interrelated spectrum of conditions that are driven by metabolism. And these include obesity, diabetes, hypertension, heart failure, kidney disease. And all of these diseases are interrelated. So what we don't understand, is really how we get, from stage 0, say here, right? So where we have no risk factors to massive cardiovascular events like this patient here, that is a depiction of a classical heart failure patient. So we really want to understand what are the metabolic drivers that lead to this. And the summary that we have right now is really this is organ crosstalk. So we see now that there is massive organ communication between the fatty tissue, adipose tissue, but then also factors such as hypertension, altered lipids, metabolic syndrome, disease of the kidneys, diabetes, and that is forming sort of like the launching pad for heart disease and cardiovascular disease that we've seen in this patient. So my lab is very much interested in this kind of intertwined, interrelated network of conditions, and we think that metabolites and metabolomics can be really a key to better understand what's going on. And we think part of the problem that we have in understanding is that we don't really understand much about the kidneys. And if I just mention you the basic physiology of the kidneys, people are usually always very amazed, because what kidneys actually do is quite amazing, and they're very altruistic. They work for the body. So kidneys have the size maybe of your fist, right? But they are perfused by 20% of every heartbeat and filter on this way 180 liters of primary urine just to make 2 liters of primary urine every day. That's like 1 ton of water every week that's being filtered just to generate your final urine every day. And the kidney does that to kind of clear the kidneys from waste, but also to keep the good stuff, right? To keep the nutrients, to keep the glucose, keep amino acids, but also remove the toxins that are not needed, and at the same time regulate blood pressure and the entire volume status. So, and we think this is really a key area that is unexplored and that we can use bioactive metabolites to provide new targets to chronic kidney disease and by extension this cardiovascular disease that we just saw. So, and I'm going to tell you a little bit about how we do that. And we do really follow this activity metabolomics approach, but we think it is really exciting that, we have this kind of entry into the body, right, to diet and microbes, exposure, exercise, and organ communication to modulate the other omics layers. So as Gary obviously coined the term. So today I show you 3 things, just give you a brief overview about our toolbox, like how we interpret mass spectrometry and combine that also with human stem cell based models such as organoids. And then I'll show you a little line of research on how hypertension can be metabolically controlled. And then 3, how organs communicate through bioactive metabolites to protect both the heart and the kidney. So, but before I do that, I have to thank the key team. So we are a bunch of people located in Germany and Denmark, and it's just a privilege for us to be able to dedicate a team and the infrastructure just to the study of kidney diseases. So it's extremely exciting, and we are very grateful for the opportunity. And sometimes we also have open positions, so please reach out if you think what we do is interesting. So I think the beauty is that with the kidney, you have obviously the serum and the urine, right? And if you measure both processes together with the kidney tissue, can do that in humans and in mice, then you have quite a good idea about what's happening in the kidney, and you can use obviously models and other bioinformatic approaches to understand what actually is happening across the kidney. You can also use the mass spectrometry, obviously, to look at metabolic amino acid modifications. What we have spent quite a lot of time on is that we need to really be able to interrogate the kidney and its transport function very controlled ex vivo, right? So we need to kind of have tools that allow us to model and understand quite in a controlled setting what's going on in the kidney. And we do that, for example, on proteomic level when we can combine just looking at single tubal mass spectrometry, just a few dozen of cells, so that we can manipulate, measure ion fluxes, or that we use microfluidics and isotopes, stable isotopes, to understand how metabolites that are put into kidney tubers ex vivo, how they are being dealt with in an injured kidney and in a normal kidney over time. And really a game changer for our laboratory were the use of organ-specific iPSC models that are allow us to model, beyond just the mouse. But we have in our lab, in collaboration with a heart group, stem cell-based engineered human heart tissues that we grow from induced pluripotent stem cells and human kidney organoids. And that's, of course, the ideal bridge to human tissue and plasma and urine samples that we get from the patients and from randomized controlled clinical trials. And we are very excited about the modeling potentials of these organoids, and I will show you later on how we can use these organoids, for example, to test for bioactivity in heart tissues. Here's an example, a recent paper from the laboratory showing that kidney organoids have quite a great impact in modeling disease. But today, I really want to go back to the metabolomics part and show you this example that was actually, again, originated from Gary's lab, where we looked at the beginning at these very simple injury models, where we just looked at this damaged kidney. Right, so this is a damaged kidney damaged by hypertension from a rat, and you can see this is there's some stuff here that doesn't belong there. But we thought, okay, what's actually happening to these kidneys before these kidneys are damaged? And we did that in this rat models and looked really at the early development of hypertension before the onset of pathology. And what we found was very surprising, because we found broad changes in amino acid metabolism that occurred before the kidney was starting to deteriorate. So we decided to, and some of these were metabolites that were known, such as leucine, which is a controller, of mTOR signaling, and we can see that beautifully in our omics data and in these models that Julio Saez did. We've also found this metabolite, the lysine, being changed in the tubers quite dramatically, and this is something we really could not make sense of. So we teamed up with various people and decided that we should try to understand this metabolism better. And we used actually a dietary labeled mouse that ate for weeks, for 1, 2, 3, 4 weeks, heavy labeled 13C labeled lysine, and using Gary's mass spectrometers and collaborating with experts such as Xavi Domingo, we were able to map essentially metabolites of lysine ranging from modified lysines to carnitines, and also a few microbial products. And what we found was that, surprisingly, the kidney was one of the major organs to participate in lysine metabolism. And it was, for some reason, the organ that modified lysine or conjugated lysine the most out of all the organs. So if you, for example, compare that to the brain didn't do much with the lysine. The kidney, for example, attached acetyl-CoA and other groups to lysine, and like this, we have quite a distinct metabolic activity in the kidney. But what's even more surprising, when we did an out-of-the-blue experiment and gave the same rats that we saw before a lot of lysine to the drinking water or through the diet, lysine as different salts, give it to male rats, to female rats. And in every case, we saw that the initial pathology, the hypertension, was blocked. Yeah? I mean, so this is extremely robust response. And this is a response that is actually stronger than, if you give, for example, a diuretic, which is quite a toxic drug. So, of course, it's an animal model, but still, we think this is a very exciting concept. What we also thought was very interesting was that we could discover new molecules based on the isotope labels. For example, here, a lysine that was conjugated with a malonyl group here. And we think this is probably formed increasingly when we add a lot of lysine to the diet, then this metabolite is being formed. And we think that maybe this could play a role in suppressing free fatty acid synthesis, and thereby, again, helping with maybe some of the symptoms of this cardiorenal metabolic spectrum of disease. And what was interesting was that essentially lysine not only conjugated to malonyl-CoA, but also to acetyl-CoA, to sugars, hexoses, and alpha-ketoglutarate, and formed these molecules that were then strongly increased in urine. So you, through the lysine, you actually generate a starvation in and the depletion of these metabolites from the body. And this is the summary of this, that by lysine, we can somehow scavenge sugar and energy equivalents, right, from the damaged kidney and excrete them. So we kind of can create an artificial metabolic sink for metabolites that maybe are not useful in this case of kidney damage. And most interestingly, this mechanism seems to be working the better the worse the kidney is. So, and I think this is a very exciting concept. So, how does this now relate to humans? We also gave a lot of lysine to humans, humans that are either healthy or humans that are at risk of developing this kidney disease and this whole disease spectrum I just mentioned. And we really can see that this has evidence for reduced damage biomarkers, surrogate markers in the urine. At least that's encouraging. And even more exciting is that now it seems to be the case that, still very controversial, but there is potential effects of giving amino acids, among them lysine, as one of the top components for kidney protection in acute intensive care settings. So we think there's really a lot of exciting knowledge about amino acid metabolism in the kidney that we can potentially use clinically.
So
The next question. Yes.
If I interrupt for a second, that last paper was really interesting. Do you think if they focused on lysine that they may have had even a more bigger impact?
Right, that's already the New England Journal of Medicine, so it's pretty good impact. But yeah, exactly, that's what we want to find out, right? So I think there is, I would suggest that the amino acid does matter. Yes.
I mean, if you just focused on lysine as opposed to all these amino acids, they may have had a bigger impact.
Right, that's what we think. Yeah,
Uh,
but let's see. Yeah, so I think this is exciting, right? Because this shows us that essentially we, by manipulating amino acid, we change, global also sugar metabolism and potentially lipid metabolism, and there is actually a known strategy for this, and that's a known strategy for exactly these patients that we have shown before, right? So these patients with a heart attack, with the kidney disease, with the diabetes, with obesity, they have this huge problems with their hearts and their kidneys, right? And there is a new treatment out that people have been developing initially for diabetes, yeah, so which is essentially an SGLT2 inhibitor. And that drug inhibits kidney sugar reabsorption, so you pee out more glucose. So it, depletes a lot of glucose from the body, but it's not really getting a ketosis, maybe a little bit, but that's a good diabetes medication, right? So that's clear, good medication for diabetes. But surprisingly, these drugs even work for patients that don't have diabetes, patients that have heart failure without kidney disease, patients that have heart failure without diabetes, patients that have kidney disease without diabetes. So by manipulating the sugar transport, you somehow can get much better outcomes in all different kinds of these patients that I mentioned before. But interestingly, no one really knows why, right? So with diabetes, you can explain, yes, you reduce the sugar. Yeah, that's helpful. Yeah, that makes sense. Yeah. But why does it work also without diabetes? And for that, we wanted to really understand that really well. And we teamed up with Volker Wallon, I think at UCSD, who has a mouse model where they just give this SGLT2 inhibitors to mice that are on a Western diet. So these are not very sick mice. They just wild type or hyperglycemic mice, and they just get the SGLT2 inhibitor for just 1 week. And we just wanted to know, where does it work? Where is it acting? And primarily it should be acting in the kidney, but maybe there were off-target effects. And we looked at all the different organs, and we looked at proteome, we looked at phosphoproteome, we looked at the untarget metabolome, and also at the microbiome. And to our surprise, we found that this drug not only acts in the kidney, but also on the gut microbiota. And interestingly, this drug acts more strongly on the wild type and not so much on the diabetic. And that's surprising, because now we see essentially that this drug changes the entire broad metabolic transport landscape in the kidney, right? And it is altering a lot of transporters there that are all involved in the recycling, uptake, and secretion of metabolites. Can see that here a little bit. Don't read at this, but there's a broad rewiring going on in the kidney. But what was very interesting was that many organic anions in the serum were decreased with SGLT2I. And these were found by target metabolomics, but what we found was that they were quite strongly reduced, particularly in the wild type, and again, not in the diabetes. And these were all different kinds of aromatic, or tryptophan-related metabolites. And we, first of all, were a little bit puzzled, because these are metabolites that are usually coming from the gut, right? And we have these excretion machineries reduced. So why do we actually have reduced amount of these metabolites? And to explain that better, I need to stop a little bit and tell you a little bit about where these metabolites come from and what they do. So many of these metabolites I just showed you are actually coming from the gut, as I mentioned, and they accumulate when kidneys fail, right? And when kidneys fail, these metabolites are thought to use their bioactivity to propagate cardiovascular disease. Some also call these metabolites toxins, although not all of them are properly toxic. So we have a reduction in these metabolites, and there's no explanation in the kidneys. So we wanted to know if the effect is happening actually in the gut microbiome. And that's what we saw in the gut microbiome. We found that there was less taxa capable of fermenting tryptophan and tyrosine. And we did even a crazy experiment. So we used a knockout mouse for this SGT2 protein and treated that with a drug. And we still saw the effect on these metabolites that are coming from the gut, which shows in our view that this, this drug has a huge effect on the gut microbiome. Okay, we asked, is that relevant in humans? So we used we used patients that have decompensated heart failure or diabetics, and we did the same metabolomics, target metabolomics assay, and we found that there is really through these drugs, also in humans, through various interventions, a reduction in these gut metabolites. And we then tested these metabolites that were regulated across the mice, the humans, in iPSC-derived engineered human heart tissues. And I will show you the potential phenotype in a second. But I would just want to remind you that these here is the patient we saw in the beginning. And these patients we looked at are these patients that have this mix of hypertension, hypoglycemia, diabetes, kidney disease. So these are exactly the patients, that we think, we cannot really treat well these days. And this is how then the bioactivity of one of these metabolites looks like. This is probably a experiment that is not physiologically relevant because the concentration of the metabolite is way too high. But what you can see here, that this little artificial heart, right, that's kind of in between two little poles, is beating spontaneously. When we add this metabolite, this little heart tissue stops beating at all, right? And when we then wait a little bit and take away the metabolite again, right, then you can see very slowly, this heart starts to beat again here, right? So there is kind of a rescue effect of this metabolite, as you can also see here. And of course, the force now is much lower. And again, this is the DMSO control. So we have really an endogenous metabolite that kind of paralyzes the heart that is being cleared through the gut's microbiome intervention. So this is how we think, we think now about organ communication. In this case, we have this SG2 inhibitors that really changes how the kidney deals with transport and toxins of these bioactive metabolites. And I could talk for 1 hour about these results and the different metabolites, but the big picture is that there is also a gut microbiome effect that we don't know now, is it direct or indirect? But for sure, we think there is communication because the kidney doesn't need to deal with these kind of toxic drugs from the gut microbiome anymore. And we think on the other side, organic anions that come from, endogenous exercise and so forth, those are increased with this drug, and this might also lead to beneficial effects. So in total, this configuration here, right, of less tubular exposure to these toxins and also the tubular transport burden of glucose will really help the kidneys. And we think that these toxins will at the end lead to a decrease in cardiovascular pathophysiology. And we identified even a marker, a GDF15 in these patients, and in the heart tissues that might give us maybe a clue if this is a relevant mechanism in those patients or not. So I conclude and I say really combining this mass spectrometry-based technologies with stem cell-derived models can really help us to understand the communication between organs better. And looking at hypertension, we can show that we can metabolically control that. And SGLT2 inhibitors, the successful drug for the cardiorenal metabolic disease spectrum that I showed you before, changes kidney configuration and microbiome to provide a favorable metabolic environment for the heart. So with this, I have to thank a lot of collaborators around the world, and Gary is for sure one of them, and Martin also very recently, because my student is just, I think, enlightened. So I'd be happy, you know, to get any questions also offline.
Okay, thank you, Marcus. Yeah, that was very impressive. I mean, it sounds like there's about 4 or 5 intertwined stories about that. For the sake of time, we're probably going to jump right into the last talk, and that is by Shua Han. And Shua is on an airplane right now, so she sent her presentation to me, and that is where I was told that this is not going to be possible. By our
If you want to introduce it, you're unmuted.
So we're unmuted right now? Okay, so Shua Han is actually an investigator at Duke University. She's a new investigator. She came out of Justin Sonnenberg's lab at Stanford. And Justin and now Shua focus largely on bioactive metabolites that are being observed within the gut microbiome. So it's a really nice segue from Martin's talk, Marcus's talk, because he basically talked a lot about gut microbiome active metabolites. So with that.
My name is Shul Han. I'm an assistant professor of biochemistry from the Duke University School of Medicine. I'm really excited here today to contribute to this AMMS lecture series. My talk today is on gut microbiome and bioactive metabolites. As many of you know, the human gut microbiome consists of incredible genetic and metabolic diversity. And it consists of trillions of microorganisms of bacteria, viral, and fungal species. So in the Han Lab, we're interested in specific species and community of bacteria and how they interact with host physiology. And specifically, many of you know that gut microbiota is associated with inflammatory responses locally in the GI tract. But it's also been shown to be associated with diverse host physiological and disease state. The ones highlighted here, such as Alzheimer's disease, cardiovascular disease, and diabetes, are actually the leading cause of death in the United States that are highly relevant to the elderly population. One disease state that's not listed here that are also highly relevant is cancer biology. There's increasing interest in investigating the interactions between the human gut microbiota and cancer. To summarize really quickly, the human gut microbiome is associated with diverse aspects of host physiology, as you have seen on the previous slide. Increasingly, we're interested in dissecting the functional roles of gut bacteria and their small molecule host physiology, and this frontier remains to be fully explored. So in terms of small molecule in the human gut microbiome, it has been shown that it's very diverse in structure, and most of them actually remain unknown. And one example shown here is the structure of butyrate. So this actually been pretty well studied and expressed in high abundance in the human GI tract, and has been shown to interact with G-protein coupled receptors, as well as modifying the chromatin state. Another structure shown here, putrescine, belongs to this class of polyamines that are highly interesting because they're relevant to aging cancer biology, and a variety of physiological responses. So I'm just giving you 2 examples of thousands of molecular structures that really remain to be fully explored. So here's the question that's really driven my postdoctoral work, as well as the current work in my lab, is to better understand what are the bioactive small molecules produced by the gut bacteria and how do they impact host biology. So if you envision the chemical space of the human gut microbiota as this pyramid, on top, there are very few that are well studied, such as short-chain fatty acid, bile acids, and several amino acid indole derivatives. And there are also remaining space we call the dark matter, but it consists of probably thousands of molecular structures that yet to be fully studied. So this actually consists of probably 2 categories of compounds. And first, the ones are identified but understudied. What I meant by that are the ones that we know the molecular structure, we know its name, but they're not really studying the context of which bacteria are responsible for their production, whether genes and pathways are responsible. And the second are the ones that are detectable. We see some kind of structure, but they're not really elucidated in a defined structure and chemical name. So those involves a lot more method development to truly understand what their structure and their functions. And then giving a few examples, polyamine pathway consists of several examples of molecule in this pathway, and each of them in bold actually has been shown to play in a diverse role in host physiology. And these are actually highly produced in the gut microbiome. So this actually captured a lot of interest over the past decade. And so what motivated us to develop this technology, in addition to better understand this interesting molecules such as polyamines, we wanted to expand our horizon to understand hundreds of pathways, how they concurrently act together. With this in mind, my colleagues and I developed this metabolic pipeline to enable small molecule discovery to study how gut bacteria interact with the host. So first of all, I just want to show you a broad overview of the methodology. And then we're going through a few examples how we apply this methodology to study gut bacteria host interactions. So here, this is described in more detail in the recent paper from our lab published in Nature Protocols. It's also a collaboration between my lab at Duke and Sundberg lab at Stanford University. So as you can see, we first compile this authentic standards of one metabolite at a time and send it through different complementary set of liquid chromatography methods running through a mass spec system and construct this reference-based library, which will guide our compound identification later on in a complex biological matrix. So give you an example, this was many years ago when we first started this project. My colleagues and I actually manually curated these metabolites based on literature search. What are the likely structures a metabolite expressed in gut bacteria culture? And now I just want to bring to your attention that increasingly there's commercially available libraries that really does this very wonderfully. So those are definitely worth your interest to pursue. And based on these internal standard collected one at a time, curated in a 96 well plate, we're able to use a complementary set of liquid chromatography method to enable us to capture the highest number of metabolites of diverse structures. And what I'm showing here, endopropionic acid, is we assign it with 2 molecular features. And this audience obviously is already familiar with the mass spec system where accurate mass in the case can respond to MZ and retention time give unique assignment. So we can use this as a reference to look at an otherwise unknown molecular structure in complex biological samples such as feces. So with this, we constructed this kind of information associated mass spec accurate mass retention time information with a pure compound one at a time. And then further, we constructed MS2 information using pure compound one at a time. So this becomes, this fragmentation pattern becomes very useful later when we are trying to validate gaining higher level of confidence in our biologically interesting metabolites. And then that's where the MS fragmentation pattern comes into place. So with this tool in mind, and then how do we next leverage this tool to really understand, to dive into metabolite profiles of diverse biospecimens. This is not limited only to bacteria, but also in the host biospecimen as well, including serum, urine, and feces and fecal contents. So here's, as you can see, in this Nature protocol, we have included detailed protocol to really do each of the steps. So right now, I'm just giving you a brief overview of what has been done. So here, with the different biological matrices, we extract this pool of complex metabolites, which then we subject them to untargeted MS1 analysis using a complementary set of chromatography method. And then for MS validation, we're only using a QC pool. QC pool is constructed using taking a little bit of every single sample in your 96 well. So you have a representative metabolites in your pool. So MS in this case is used to validate biologically interesting metabolites and its structure. And then next, for data analysis, we use this interface called MS-Dial, which you can use a variety of different user interface that analyze these. And then this software enables us to do featureization, identification, and elimination based on QC controls. And next, describe in more detail, share on GitHub, basically it's described more in detail in the Nature protocol paper. In brief, and this pipeline is constructed to enable us to join experiments, different experiments, and filter out ones we don't want or normalize data across experiments, so then we can do biologically interesting analysis. So next, based on this tool we have built, I want to show you a few vignettes and how we can apply these tools to diverse application of gut microbiome studies. So first, we have used this tool to construct a collection of in vitro and in vivo database. Basically, the in vitro, we surveyed 170 plus prevalent human gut bacteria strains. And so these are grown in single bacterial culture in vitro to understand what are the specific metabolic markers associated with distinct human species. And then for the in vivo context, we also express a single bacteria species or the community of bacteria species in otherwise germ-free mice. So these mice, then we harvest biofluids such as feces to represent local production in the GI tract, and then serum and urine to represent circulation, metabolite produced in the circulatory system. So these collectively will provide us really exciting input, and which I will give you a few examples on the next few slides. Now, I'm showing you a heat map summarizing 150 bacterial species that are grown in single in vitro culture in a very rich undefined media. So everybody's growing the same media to enable cross-species comparison of metabolic profiles. And these ones are clustered based on their 16S sequencing, so they're clustered close to its own phyla, where they come from. And here are the 5 diverse phyla that are representative of the ones that are present in the human gut microbiota. And you can see that just looking across this broad spectrum of data, there are, phylum-wide conservation of production, as evidence here in red, which indicate production relative to media control. And there are also phylum-wide conservation of consumption of specific metabolite. Now, when you look more deeply into one metabolite at a time, for example, tyramine, which is a metabolite relevant to neurological behavior in mammals, if you look across the production profile of this 158 bacterial species and strains, and most of them actually are not really producing much. right, compared to its control. But except therefore, Enterococcus faecalis and Enterococcus faecium strains and species are actually high producers. And for another example, pantothenic acid and vitamin has been shown to associate with IBD patients. So looking across this 150 bacteria taxa, most of the species actually are not really doing much with this metabolite, whereas 4 strains of Clostridium cadaveris seems to be high consumers that deplete this metabolite significantly. So this is just one example of things we can really learn from this metabolic data. What are the unique biological markers of these gut bacterial species, right? What are the conserved features across broadly conserved across phylum? And going back to the polyamine pathway that I mentioned earlier, these guys are actually highly produced by the human gut microbiota. And these are particular of interest because each of the one highlighted in bold here are actually relevant to cancer biology, aging, cellular replication. So what my interest here is what are the, what contribute to their metabolic variations? Who's producing them? If it's a bacteria-specific phenotype, what are the genetic basis for such metabolic variations? So to address this question, I'm leveraging the existing data we have, I showed you on the previous slide, the metabolic profiling, one strain at a time, and then combining with comparative genomics of the annotated genomes of these associated strains. So in this case, I'm showing you actually the same metabolic map that I show you on the heat map, except that I'm now focusing on specific metabolite of interest and presenting in a slightly different way. And you can see, first of all, the phylum borders, the 5 phylum are shown here in different colors. And then I will first focus on the production of ornithine and citrulline as shown here. And as you can see here, there are actually clusters of firmicutes that are high producers of ornithine and citrulline, as shown here. And if you look across this phylogenetic tree, and the one shown here, a few actinobacteria are also high producers of ornithine and citrulline. This is a little unexpected because, as you can imagine, 6S tree, they're really far apart. So they're not really evolutionarily connected. And yet they share metabolic similarity. So, and then I ask, okay, so what are the genetic bases really contribute to these kind of production? What I find this ARC gene cluster is actually present across these firmicutes that express high-level ornithine citrulline and also detected here as well by homology search. So this one taught us that phylogenetic relationships does not always predict metabolic functions. And looking at the same set of data, but different data points, now let's focus on putrescine and augmentine production here in different shades of green. And we find that here's a cluster of proteobacteria that are high producers of putrescine and augmentine. And then when you look across this phylogenetic tree, there are also conserved production among the fusobacteria as well as the firmicutes. And this is very interesting because, and then I dig into the genomics to really confirm, is this also conserved as well? And then for the speed genes, actually well studied in E. coli, and this is actually conserved across these proteobacteria. But then when I search further, it's actually not detected by homology in fusobacteria and firmicutes, which suggests that these has other mechanisms in gene cluster responsible for these metabolic productions. So to summarize, while metabolic capacity is shared, the genetic basis for this capacity is not always conserved. So taking everything together, coupling comparative metabolomics and genomics, as we did here, presents a powerful tool to review conserved and unexpected gene to metabolite relationships. So now I showed you a whole ton of in vitro cell culture data. So can we leverage some of the knowledge here to manipulate metabolite production in mouse models? So in this case, I'm going to show you a proof of principle experiment using one of my favorite metabolites, augmentine, which is a aging-related metabolites that confer neuron protection in mammals. So I wanted to toggle the production of augmentin in the mouse, not only locally in the gut, but also systemically in the host. So what I do here is I use the high producer of augmentin that I identify from my in vitro screen called Citrobacter petuclensis. Then I use this strain and express in otherwise germ-free mice. So these germ-free mice are now colonized with this bacteria species. And then what I see locally in the GI tract, in the feces and fecal content, I see a high level, this is log 2-fold change, so it's 6 to 8-fold increase of augmentin locally in the GI tract, and also systemically as detected in urine. So this demonstrates to us that by kind of manipulating gut membership and introducing high-producing members into the community may actually help us modulate the expression of these metabolites locally and systemically. So now another interesting question in the field oftentimes is it's a fairly complex community. How do we identify metabolic impact of individual microbes that contribute to this complex community? So to address this question, we use the 6-member defined community that represent diverse range of metabolic production across phyla. And then we also chose 2 individual colonization, including a B theta from Bacteroides phylum, and C sporogenes representing phylum Firmicutes. So the Bacteroides and Firmicutes actually are the top 2 phyla represented in the Western gut. And then we chose B theta and C sporogenes because there are actually very few that are genetically tractable. So we actually can manipulate their genome to really study how to manipulate metabolite production if needed. So the experiment design, as I described earlier, we have 3 different conditions and cohorts of mice. One consists of 6-member defined community, and the other ones are monocolonized, meaning that they're just having expressing one gut bacteria at a time, in this case, C sporogeny on the left, and B theta on the right. What we find using serum metabolomics, because we're interested in the metabolite end up in the circulation, which has potential to reach diverse tissue and cell types. We saw, even though there are diverse set of metabolite detected in the 6-member defined community, and several of them are highly specific and can be traced back to the unique bacteria that produce them. To express this in slightly different ways, as you can see, for several serum metabolite detecting C sporogeny expressing mice, many of these are detected in high ion count only in C sporogeny, but mice with C sporogeny, but not in the germ-free mice or the mice with B theta. And consistently, for the B theta specific metabolites such as indoxyl sulfate is highly produced only in the serum of mice that are associated with B theta, but not in the mice that express C sporogeny or germ-free control. So just to summarize, I showed you a development of technology that's enabled for a broad range of small molecule discovery in the context of the gut microbiome. And several vignettes or examples demonstrating in vitro culture, in vivo studies, how we can apply this technology to study diverse goals in gut microbiome sciences. And lastly, in summary, I just want to emphasize that our tool is really broadly applicable to identify small molecule mediators of gut bacteria host interactions in health and disease. And in the end, I want to give a quick shout out to my lab, which we just celebrated our 2-year birthday at Duke University. Specifically, we use chemical and genetic approaches to decipher the chemical language of gut bacteria in host aging and longevity. Thank you so much for your attention. I hope you enjoyed this presentation, and thank you.
Yeah, so I think we're all set now with the series of talks and course material. I'll probably end it there, so all of you can get back to your normal day. I really appreciate the attendance of from the beginning, roughly 1,300 attendees to today. It ended up at about 500. Started off with about 570. So it really, I think, speaks to the interest level and
what we have in terms of metabolomics and the technology moving forward. And I really appreciate Marcus, your time, Martin's time, and Shua for putting together this presentation, which really felt like she was here. So I can't believe that we were able to pull that off, even though we were told that we were not able to. So thank you very much. And